Glucose is yeast's favorite food because they can get more energy out of glucose than any other sugar. In a Nature paper, our group at UC San Diego discovered a new way that yeast makes sure it gets as much glucose as possible. If yeast sense glucose in their surroundings, a complex genetic pathway is activated that shuts down the metabolization of all other types of sugar. In our study, we examined the dynamics of this process. We discovered new strategies that yeast employ to maximize glucose consumption when both glucose and yeast's second favorite food, galactose, are present. In our bioengineering lab at the Jacobs School of Engineering, we grew yeast in an environment with a constant level of galactose. Then, we periodically added temporary bursts of glucose and measured how the cells responded. In this movie, we have two panels. On the left are time-lapse images taken with a microscope. As time passes, we can see two things occurring. First, the red increases as glucose enters the environment, and then the red decreases when glucose levels drop. Second, the amount of green in the yeast cells changes over time. The green is a specialized genetic marker which tells us the activity of the galactose metabolization pathway. Since there is always galactose in the system, the cells will eat galactose whenever glucose is absent. When this occurs, the cells begin to glow green. However, as glucose is pulsed into the system, the cells react by turning off the galactose pathway in favor of the glucose pathway. With the human eye, it is difficult to tell exactly what is happening, so we use computers to examine these microscope images and measure the precise activity of the galactose pathway. These data are shown in the right panel. On the right, we see two curves evolving in time. The first is a red curve that reads off the exact amount of glucose in the chamber at any given time, and we have periodically varied this level to create a sine wave. The green curve shows us the average activity of the galactose pathway in the cells as a function of time, and the shape of this curve is what we are interested in. As time increases, we see a general upward trend in the activity of the galactose network. However, as glucose goes in and out of the chamber, peaks and troughs are created on top of the upward trend due to the glucose pathway turning off galactose activity. By doing these experiments over and over again at different frequencies, we gain insight into the dynamics of the glucose-galactose interaction. We compared our experimental results to existing computational models of these pathways and found that the current understanding was incomplete. The discrepancies between the experiments and the models eventually led us to the discovery of a new form of regulation between the two genetic pathways. Specifically, as the glucose pathway is turned on, it creates a chemical signal which actively breaks down and thereby inactivates the messenger RNA created by the galactose network.